Now we will talk about the immunity, girls. In the last video, we talked about producing the antibodies from the B cells. We said this process needs a few days until the specific antibodies are produced. Finally, inshallah, the virus or the pathogen will be overcome, inshallah. This process needs time, but also there are other types of the T cells, and the B cells will be produced finally. What are these cells? They are memory cells. Memory cells. These memory cells will store all the information about the pathogen. Will store all the information about the pathogen. So this will give us immunity against the disease if it comes, or against the pathogen if it enters again. Okay, so let's give the definition of the immunity, then explain it again. Immunity is the ability to resist or to recover from an infectious disease. What is infectious disease? Infectious disease is a disease that is caused by a pathogen such as virus or bacteria, and this infectious disease can be passed from one to another infection. Okay? Now, this infectious disease, when it happens once, okay, the process of producing the T cells and the B cells will take place over about 5 to 7 days mostly, to produce the needed antibodies. So it takes long time. If sometimes as what we said in the last video, if the immune system is weak, or if the body of the patient is not healthy, not strong enough, maybe the pathogen can kill this patient sometimes. Okay, but if inshallah the patient survive, or if the viral infection or the bacterial infection wasn't fatal, يعني wasn't a killer infection, this infectious disease will cause the production of memory cells. The production of memory cells. Okay, as what you see here, at the end of the process, some memory cells of B cells, also of T cells, will stay inside our blood. As long as we live, will stay inside our blood. طيب, what for? What for? I will tell you what for. The next time, if this pathogen thinks to enter to your body, these memory cells directly will produce the needed amount of killer T cells and B cells that produce the antibodies directly, without needing the 5 or 7 days. No, directly. خلاص, they kept it in their memory. They have all the information about this pathogen. So these antibodies will be produced directly. طيب, if the antibodies are produced directly, what will happen? The virus will not have any chance to infect any cell. this virus will be killed directly will be destroyed from the memory cells that were inside our blood previously. That's why, girls, most of you know, if you had chicken pox once, you will never have it again. Right? If you have it once, you will never have it again. Most of the bacterial infections also are the same, exactly the same as this pattern. Follow this pattern. From here also is the idea. Okay, before let's give only some definitions. We say, what is the immunity? Immunity, we said it is the ability to resist or to recover, and it can be passed from the mother to the fetus. What is the fetus? The fetus is the baby during the pregnancy in the uterus of the mother. Also, it can result from being infected with a disease or from being vaccinated. The idea of the vaccines come from what I explained to you. When some scientists, it has also stories if you like to read in the history how they discovered the vaccine, and this is nice if you want to search about it. Some scientists, some doctors observed, and they found that some patients, if they have the disease once and they were in contact with patients for the second time, they will not be affected. They will not be infected with the pathogen. Then they search, they take from the blood of these patients, and they found that they have memory cells, and from here they start in the idea of vaccination. They said if the pathogen make these memory cells, if it was enter to the body normally, why don't we enter this pathogen and get the memory cells from its entrance? طيب, you will ask me how they enter the pathogen to the body. Then we will be infected, we will have infection, right? I will tell you that they work on killing the bacteria, if it was bacteria طبعا, because the bacteria is a living thing. They kill this bacteria, then they inject it to the body. Your body will work on the bacteria without knowing that it is a dead cell. It is not your immune system will work on the antigen of this bacteria. The antigen of this bacteria. Once the antigen is entered to the body, the immune system will go through the same process. The macrophage will destroy the bacteria. Then the antigens will be released. The T cells will activate the killer T cell and activate the B cells and make memory cells. That's what we want. خلاص, memory cells are ready. Next time, if we are infected with a real or live bacteria from the same type, our memory cells will work on it. We will not have the infection. This is the vaccine. This is the idea of the vaccine. From the virus, you will tell me, طيب, the virus is not a living thing. How it can be killed? They work on it to make it inactive. Okay, They work on it in the labs. They make specific processes to inactivate the virus. Then the virus will be entered to the body, and the process will take place to produce the memory cells. That's what we want. Producing the memory cells is the aim of the vaccine. So it is, the vaccination is a substance prepared in the labs from killed or weakened pathogen. So even the bacteria, sometimes it is weakened, not killed, that is introduced into the body to produce immunity, as what you see here in the injection. What are these? This is a killed or weakened pathogen. When the pathogen, sometimes they take part only the antigen. Sometimes they take only the antigen from the virus, only the antigen from the bacteria. Finally, the antigen will be entered to the blood. This will cause the production of the memory cells. The memory cells will be ready. Once the real virus, as what you see in this picture, the real strong virus enters to the body, the antibodies are ready. It can't do anything. So this is the very important issue that all the world now is competing to have. All the world countries, especially the very advanced countries, they are competing to be the first ones to produce the vaccine for the coronavirus. Okay, because they will save the life of the world, the lives of millions of people. Okay, let's see. Here I want just to show you the T cells, how from the pathogen, this is this is an infected cell, and on the surface of the infected cell are the antigens. These antigens will activate on the T cells specific proteins, matching proteins. Then the helper T cell will activate the killer T cells and will activate the B cells. Look at the B cells. B cells also will produce, will produce the specific or the matching antibodies. Here they show how the antigen enters to the cell, then releases the antigens on the infected cell. Then this will activate the T cell. Then the T cell will activate the B cell. The B cell produces the antibodies, matching antibodies. Finally, billions of T cells and B cells will be produced. At the end, memory cells will be produced. Here, this is what I want to show. Memory T and B cells with this specific antibody, matching antibody. Because as we said, every antigen has a specific antibody, like a lock and a key. Okay, so we can't have the same B cells for all pathogens. Every pathogen has a specific antigen which will have a specific antibody. That's it. We will watch together now a small video about the vaccination, example about influenza vaccine, how they prepare it. They prepared it in the lab to take from the influenza virus only the antigens. Okay, then in a small amount, then they enter the antigen to the body. The antigen is not like the virus. It can't replicate. It can't make the same effect of the virus, but it will give the same stimulation. It will stimulate the immune system the same as the virus. It will have the same effect. Let's see how it will work together.
The immune response. How vaccines work. Influenza vaccines are able to trigger an immune response by mimicking viral infection. They are usually manufactured using inactivated or killed virus particles taken from various circulating influenza strains. Inactive fractioned viral components contain the subvirion particles hemagglutinin, HA, and neuraminidase, NA. In Canada, influenza vaccines are administered. Fractioned particles containing the foreign antigens hemagglutinin and neuraminidase are released from the vaccine into the bloodstream. There, they are met with an immune response mediated by.
Now, as what you see from the injection, what were entered only these antigens. The HA, they talk about it, small fragments from the virus. These fragments or these particles are the antigens. They enter them directly. Macrophage will come. This is the first thing.
By various immune cells, including macrophages, T lymphocytes or T cells, and B lymphocytes or B cells of hemagglutinin, breaking it into smaller components. After
the macrophage swallowed the antigen, then the antigen will appear.
Ingestion, macrophages display the hemagglutinin antigen on their surface in combination with a specific receptor known as the major
Specific receptors for the antigen will appear on the surface of the macrophage. What will happen now? What will be stimulated? The T cells.
Compatibility with the MHC.
The T cell with
Upon binding to the MHC receptor, T cells become activated and proliferate into either cytotoxic T cells. Regulatory activated helper T cells express hemagglutinin receptors specific to the vaccine strand.
Also,
unlike T cells, B cells are able to ingest hemagglutinin independent of the MHC. Once internalized, B cells process the hemagglutinin antigen and present it on their surface in combination with an MHC. When activated helper T cells interact with activated B cells expressing antigen MHC receptors, they begin secreting lymphokines which have several effects. Lymphokines trigger activated B cell proliferation, which leads to either their differentiation into memory B cells or into plasma cells. Plasma cells produce hemagglutinin antibodies specific to the strain of influenza contained in the vaccine. Memory B cells aid in future immune response when exposed to an active influenza virus. When an infected host sneezes towards an uninfected person, the nasopharynx is exposed to aerosol droplets containing whole, live influenza virus. Once inhaled, the influenza virus attempts viral colonization of nasopharyngeal epithelial cells. Assuming that the vaccine strain matches that of the live virus, hemagglutinin antibodies block viral attachment of live influenza virus to.
You see here, this person had the vaccine. Directly the virus antigens were blocked. You see how they are blocked by the antibodies directly because the memory cells exist naturally in his
Most epithelial cells, and overall disease is either avoided or diminished in severity.
So, no work, nothing. The virus couldn't work in the patient's body. That's it, and that's what we need, inshallah. We are waiting for them to make this vaccine, inshallah. Yalla, see you in the next part.